Hello friends, welcome to my Microbe channel. Today we have came with the topic that is acid fast staining. So what are acid fast bacteria? Those which have high content of mycolic acid in their cell wall are known as acid fast bacteria, and also they have large amount of fatty acid, wax and complex lipid in them. Due to the waxy cell wall component, Mycobacterium are acid fast and they can retain the carbon function after rising with acid solvent, and also they are highly resistant to disinfectant. So what is the history about acid fast staining? It is a differential staining technique. It was first developed by Ziehl and later it was modified by Neelsen. So, this method is also known as Ziehl Neelsen staining technique. Neelsen in 1883 he used this carbon function and heat then decolorized with acid alcohol and counted stain with methylene blue. So this method it is also known as Ziehl Neelsen staining technique because it was developed by this both scientists. So the next is, what is the aim of this staining? So the aim of this acid fast staining is that it can differentiate acid fast bacteria from non-acid fast bacteria. So the principle of acid fast staining. As you know, several kind of bacteria they contain mycolic acid and waxy material in their cell wall. But some bacteria do not get stained with simple staining or ordinary staining technique, that is gram staining. So the chemical composition of the cell make them impermeable to the stain. So if such cell it is stained with Ziehl Neelsen carbon function along with the steaming, this cells get stained. So after that, when a strong decolorizer, that is acid alcohol, when it is added to the stain, the non-acid fast cell loses the stain and automatically they become colorless. The application of this counter stain methylene blue, it is responsible to stain this colorless cell, and the result of this is acid fast bacteria they appear pink in color while non-acid fast appears blue in color. The procedure: so firstly take a clean grease free slide, prepare a smear of given bacterial suspension. After that, float the smear with carbon function stain. At the same time, hold the slide on boiling water bath for 5 minutes. If the slide is dry, add more stain on it. After that, cool the slide for at least 5 minutes. Then add acid alcohol decolorizer on it, and after that add counter stain, that is methylene blue, for 1 minute. Then wash the slide with tap water, allow the slide to dry and observe the slide under low power objective lens or high power objective lens. You can use 10x or 14x, or you can use 100x oil emulsion lens. So this staining technique it is mostly useful in the diagnosis of tuberculosis, which is a causative agent of Mycobacterium tuberculosis, and it is a acid fast bacteria. So this was all about acid fast staining. Thank you. Please like, share and subscribe my channel.